Good day everyone. We are group 1 and we will be tackling about the metabolism of carbohydrates to know its process, stages, importance, and overview of the study. So what is the metabolism of carbohydrates? The metabolism of carbohydrates is a fundamental biochemical process essential for energy production and physiological stability in living organisms. In this exploration, we will trace the journey of carbohydrates as they break down into glucose, examine glycolysis and the Krebs cycle, and highlight the pivotal role of ATP production. So the first major stage in carbohydrate metabolism is glycolysis. This process occurs in the cytoplasm of cells and involves the conversion of glucose into pyruvate, generating a small amount of ATP and NADH. Glycolysis is crucial because it provides both immediate energy and the necessary building blocks for other metabolic pathways. Following glycolysis, pyruvate enters the mitochondria and is converted into acetyl-CoA, which then proceeds to the Krebs cycle, also known as the citric acid cycle. This series of reactions further breaks down acetyl-CoA, releasing carbon dioxide and transferring energy to carrier molecules such as NADH and FADH2. The final stage of carbohydrate metabolism is ATP production through oxidative phosphorylation. During this stage, the NADH and FADH2 generated earlier donate electrons to the electron transport chain located in the inner mitochondrial membrane. This process creates a proton gradient which drives the synthesis of ATP, the primary energy currency of cells.
In carbohydrates, it is important to know the essential macronutrients that serves as the body's primary energy source. They are broken down into glucose, which fuels various bodily functions, particularly brain activity and physical performance. Additionally, carbohydrates are stored as glycogen in the liver and muscles, providing a reserve energy source during fasting or exercise. Complex carbohydrates, such as those found in whole grains, fruits, and vegetables, release energy gradually, helping to maintain stable blood sugar levels and prevent energy crashes. They also support digestive health by providing dietary fiber, which aids digestion, regulates appetite, and promotes a healthy gut. Furthermore, consuming enough carbohydrates prevents the body from breaking down proteins for energy, allowing proteins to focus on crucial roles like tissue repair. A balanced intake of carbohydrates is essential for sustaining energy and overall well-being.
Digestion begins in the mouth where carbohydrates are greeted enthusiastically by an enzyme called salivary amylase secreted by the salivary glands. As we chew our food thoroughly, this enzyme starts breaking down starches into smaller molecules like maltose. This is why bread or rice may taste slightly sweet after a bit of chewing because maltose is a sugar. However, this step is relatively brief as the food quickly moves to the next phase. After being swallowed, the partially digested carbohydrates travel down the esophagus and into the stomach. Interestingly, the acidic environment of the stomach temporarily halts carbohydrate digestion because the low pH deactivates salivary amylase. However, this pause is only momentary as the real action resumes once the food moves into the small intestine. As the partially digested food, now called chyme, enters the duodenum, which is part of the small intestine, it is met by the pancreas' contribution to digestion, which is the pancreatic amylase. This enzyme energetically continues breaking down starches into maltose, a disaccharide. At this point, carbohydrates are broken down into simpler forms, but they're still not quite ready for absorption. The final breakdown occurs in the small intestine's lining, where specialized enzymes like maltase, lactase, and sucrase, secreted by the intestinal walls, efficiently break disaccharides, such as the maltose, lactose, and sucrose, into monosaccharides, glucose, galactose, and fructose. These are the simplest forms of carbohydrates and are now small enough to be absorbed. For the absorption of these carbohydrates, the small intestine, specifically the jejunum, plays a starring role here. The monosaccharides are absorbed through the intestinal lining known as enterocytes into the bloodstream. Glucose and galactose are absorbed actively via sodium glucose transporters, while fructose is absorbed passively through facilitated diffusion through GLUT5 transporters. Once in the bloodstream, these monosaccharides are transported straight to the liver via the hepatic portal vein. In the liver, fructose and galactose are converted into glucose, which becomes the body's primary energy source. The glucose is then distributed throughout the body or stored as glycogen in the liver and muscles for later use. To summarize, the digestion and absorption of carbohydrates is a highly coordinated process that begins in the mouth and is completed in the small intestine. It involves several enzymes and mechanisms to ensure carbohydrates are efficiently converted into glucose, fueling our body with energy.
Glycolysis is a primary source of energy and carbohydrate metabolism. It is related to energy production in cells. The metabolic pathways of glucose take place in the cytoplasm of the cell. It is the first step in the breakdown of glucose, a 6-carbon sugar, to extract energy for the cell. This process occurs in both aerobic and anaerobic conditions and is vital for ATP production, which powers various cellular activities. During glycolysis, a molecule of glucose is broken down into 2 molecules of pyruvate, releasing energy in the form of ATP and NADH. These products can then be further utilized in cellular respiration or fermentation, depending on the availability of oxygen. The significance of glycolysis has a role in the primary pathway for breaking down glucose to produce in the form of ATP. This is essential because it provides quick energy that generates ATP without the use of oxygen during anaerobic conditions. It also initiates cellular respiration, which is the first step in glucose metabolism, which feeds the citric acid cycle and oxidative phosphorylation in energy production. Thirdly, this provides energy without mitochondria, as in this process, it supports all cell types. Lastly, supplies other biosynthetic pathways supporting the synthesis of macromolecules. Moving forward, I will explain to you the key inputs and outputs of glycolysis. The ATP has a net gain of 280 molecules per glucose molecule because before having a net gain of 2 ATP, it has 4 ATP produced, which will be minus the 2 ATP used. So therefore, 280 molecules is the net gain result of the NADH has 2 molecules of NADH being produced where it can be used in the electron transport chain to generate more ATP during aerobic respiration. Pyruvate has 2 molecules of pyruvate are produced per glucose molecule, where pyruvate can be further metabolized as aerobically, which means enters the mitochondria for conversion into acetyl-CoA and further oxidation in the citric acid cycle. Next is the anaerobically that converted to lactate in animals or lactic acid fermentation or ethanol and CO2 in yeast or alcoholic fermentation. Water. Small amounts of water are produced as byproduct. Overall reaction of glycolysis. Glucose plus 2 NAD plus 2 ADP plus 2 pi equals to 2 pyruvate plus 2 NADH plus 2 ATP plus 2 H2O.
Before delving into the actual process, I will first describe each phase of glycolysis, which are the first and second phases. The first phase is the investment phase, which consumes energy. Moreover, by the end of this phase, it will consume 2 ATPs. The second phase, it is otherwise known as the payoff phase, wherein it involves the production of energy. Ultimately, this phase will contribute to the results of 4 ATPs and 2 NADH. With everything explained, each phase has 5 steps, thus resulting in 10 steps overall. Now let's proceed to the first step, which originates in the first phase. In the first step, this step starts off with a glucose molecule. The hydroxyl group from the glucose molecule is replaced with a phosphate group coming from an ATP molecule, losing a phosphate group and transforming into ADP with the help of the enzyme hexokinase, which catalyzes the said reaction along with the magnesium 2 plus ion that reacts with it. The said phosphate group is attached to carbon 6 and is then named glucose 6-phosphate with the process of phosphorylation. In relation to the concept of the delta G value, it is an irreversible reaction, thus it cannot be undone. In the second step, with the glucose 6-phosphate from the first step, it then becomes fructose 6-phosphate facilitated with the magnesium 2 plus cofactor. Glucose and fructose are known to be isomers of each other. Thus, the enzyme that aids and is responsible for the said reaction of rearranging is the isomerase enzyme, specifically the phosphohexose isomerase. About the concept of the delta G value, the reaction is reversible. In the third step, with the fructose 6-phosphate from the previous step, it is then transformed into fructose 1,6-biphosphate through phosphorylation. The hydroxyl group from the fructose 6-phosphate was replaced with a phosphate group. Thus, in the product side of the reaction, it contains two phosphate groups. Similar to step 1, the phosphate comes from an ATP molecule when it loses a phosphate group. It then transforms into ADP with the help of the enzyme phosphofructokinase, which catalyzes the said reaction, and the magnesium 2 plus ion that reacts with it. With the reaction's delta G value, it is said to be irreversible. For the fourth step, the cleavage of fructose 1,6-biphosphate. Ketone and aldehyde are found in the product side of the reaction. 3-carbon aldehyde with hydroxyl group is otherwise known as glyceraldehyde. A phosphate group is also found in carbon 3, thus is called as glyceraldehyde 3-phosphate. The hydroxyacetone phosphate, in which is a 3-carbon ketone otherwise known as acetone. With the reaction's biochemical delta G value, it is said to be reversible. With such being indicated, the reaction is catalyzed by the enzyme aldolase, which is used to catalyze the reversible aldol condensation reaction. In step 5, this is the last step of the investment phase. The dihydroxyacetone phosphate from the previous step is converted into glyceraldehyde 3-phosphate. This is a reversible reaction. The chemical relationship between the reactant and product is that they both have 3 carbons, 5 hydrogen atoms, 6 oxygen atoms, and a phosphorus atom. Because of them having the same number of atoms but a different structure, these are called isomers, a rearrangement reaction catalyzed by the enzyme isomerase. Focusing on the substrate molecule 3-carbon ketose sugar or ketose sugar as is, with a phosphate group attached to it. The enzyme needed to catalyze this reaction is the triose phosphate isomerase. Step 6 is the beginning of the payoff phase. This is a reversible reaction. It has glyceraldehyde 3-phosphate in the reactant side, and in the product side of the reaction, 2 phosphate groups are attached to. Thus, the product of the reaction is 1,3-biphosphoglycerate through the conversion of NAD plus into NADH, with an H plus ion produced through a reduction reaction with the enzyme glyceraldehyde 3-phosphate dehydrogenase, as it oxidizes a substrate by removing hydrogen and at the same time reducing NAD plus to NADH. Take note that everything is multiplied into 2 as there are 2 molecules of glyceraldehyde 3-phosphate that enters the phase, meaning 2 NAD plus is reduced to 2 NADH. In step 7, we are able to gain 2 ATP molecules. We know that the reactant is 1,3-biphosphoglycerate. In the reaction in the reactant side, we lose a phosphate group, having no phosphate group in carbon 1, and it is only present in carbon 3 in the product side. Thus, it leads to 3-phosphoglycerate as our product. With the lost phosphate group, it was transferred to a molecule of ADP to produce ATP with a process of substrate-level phosphorylation with the enzyme phosphoglycerate. As we're still in the payoff phase, everything is multiplied by 2. Thus, we have 2 ADP and 2 ATP. In step 8, as we have 3-phosphoglycerate from the previous step, our product is then 2-phosphoglycerate as the phosphate group has been shifted from position 3 to position 2 by the enzyme phosphoglycerate mutase, a type of isomerase enzyme that moves a functional group from one position to another. In step 9, in this reaction, we lose a hydrogen and a hydroxyl group. Therefore, we're removing water out of the equation and becoming a side product of this through a dehydration reaction. With the 2-phosphoglycerate, it then becomes phosphoenol pyruvate with the enzyme enolase that catalyzes the reaction as it proceeds through an enolic intermediate. Thus, in this step, we have the dehydration of 2-phosphoglycerate into phosphoenol pyruvate, gaining 2 water molecules as there is 2 glyceraldehyde 3-phosphate molecules starting with step 6. For the last and final step, step 10, we are able to gain another 2 ATP molecules. The phosphoenol pyruvate is then converted into pyruvate with the needed cofactors, which are the magnesium and the potassium ions, with the enzyme pyruvate kinase that catalyzes the reaction as we are losing a phosphate group. With the phosphate group that we've lost, it was transferred to a molecule of 2 ADP molecules to produce 2 ATP molecules. As stated earlier, that everything is multiplied into 2, which is why we have 2 ADP molecules converted to 2 ATP molecules. Thus, overall in this phase, we have generated 4 ATP molecules.
Glycogen synthesis. This is known as the glycogenesis. Here we'll discuss the process formation of glycogen from glucose. The process of glycogen synthesis of glucose is converted into glycogen for it to be stored, where they are stored in the liver and muscle cells. It maintains an energy reserve in the body that can be used during periods of low blood sugar or increased demand and energy. The main points of glycogen synthesis are, first, purpose in storing the excess of glucose as glycogen, where it can be broken down into the glucose when the body needs energy. Secondly, the location where it is stored in the body. Steps of glycogen synthesis. Phosphorylation of glucose. Glucose is phosphorylated to glucose 6-phosphate or G6P by exokinase or glucokinase. Isomerization to glucose 1-phosphate. Glucose 6-phosphate is converted to glucose 1-phosphate or G1P by phosphoglucomatase. Activation of glucose. Glucose 1-phosphate reacts with UTP to form UTP glucose, catalyzed by UDP glucose pyrophosphorylase. Addition to glycogen chain, glycogen synthase adds glucose from UDP glucose to the existing glycogen chain, forming A1,4 glycosidic bonds. Branching of glycogen. The branching enzyme introduces A1,6 glycosidic bonds to create branches in the glycogen molecule, increasing solubility and accessibility. Significance of glycogenesis. Energy storage. Glycogen serves as an energy reserve that can be quickly mobilized during periods of high energy demand. Blood glucose regulation. In the liver, glycogen can be broken down to release glucose into the bloodstream to maintain normal blood sugar levels. Gluconeogenesis versus glycogenesis. Gluconeogenesis is different from glycogenesis. It is the metabolic pathway through which glucose is synthesized from non-carbohydrate precursors such as lactate, glycerol, and certain amino acids, primarily in the liver and kidneys. This process is crucial during fasting or starvation to maintain blood glucose levels. While glycogenesis, on the other hand, specifically refers to the formation of glycogen from glucose 6-phosphate and not gluconeogenesis.
Converting the energy from a proton gradient into ATP. It is also the cell's main energy source. So, proton gradient. ATC role or electrotransport chain pumps proton into the intermembrane space, creating a high concentration of protons outside the mitochondrial matrix. And also, proton motive force. Proton motive force, this is the gradient, a form of stored energy, drives ATP production within the gradient. So, ATP synthase mechanism, it is a proton flow back into the matrix through ATP synthase. This flow rotates the ATP synthase, converting ADP and PI into ATP. ATP yield. So each NADH yields 2.5 ATP, and each FADH2 yields 1.5 ATP, a total yield of 28 to 30 ATP per glucose.
The importance naman of the oxidative phosphorylation is that the chemiosmosis ensures efficient ATP production, powering essential cellular activities. The PPP, or also known as the pentose phosphate pathway, occurs in the cytoplasm and provides NOD-PH and ribose 5-phosphate for biosynthesis and antioxidant defense. There are phases the phase 1 is the oxidative phase, the NOD-PH production. The glucose 6-phosphate is oxidized, generating NOD-PH for fatty acid synthesis and cellular defense against ROS. The phase 2 or the non-oxidative phase, the ribose 5-phosphate, converted from ribulose 5-phosphate for nucleotide synthesis. For the sugar interconvention, it produces other intermediates for cellular needs. The functions naman for NOD-PH, it supports biosynthesis and acts as an antioxidant. For the ribose 5-phosphate, it is essential for DNA and RNA synthesis.
Hello there, listeners. Let's talk about something really interesting today, like how your body keeps your blood sugar in check. It's all thanks to some key hormones that work together like a team. We're talking about insulin, glucagon, and a few others like cortisol, epinephrine, and growth hormone. These hormones are constantly adjusting things to make sure you have enough energy, but not too much. It's kind of like your body's way of playing the Goldilocks game. Not too high, not too low, but just right. First off, what is blood sugar? Blood sugar, or glucose, is the energy your body uses to fuel everything from your brain to your muscles. After you eat, your body breaks down food, especially carbs like bread or pasta, into glucose, and that enters your bloodstream. Your cells then use it for energy. But here's the catch. Too much glucose in your blood, not too good. Too little glucose, even worse. So, your body needs a way to balance it, and that's where hormones came in. Now, the main player here are insulin and glucagon. Think of them like the best friends who help keep things in balance. Insulin is the good guy here. After you eat, your blood sugar rises, so insulin comes in to help. It's like your body's sugar controller, telling your cells, hey, grab that glucose and use it for energy. Insulin also helps your liver store extra glucose for later, kind of like putting leftovers in the fridge for the future. So insulin's job is to lower your blood sugar after meals. Glucagon is the backup hero. When your blood sugar drops, maybe you haven't eaten for a while, glucagon tells your liver, time to release that stored glucose. It makes sure your blood sugar doesn't dip too low, keeping everything steady. So, glucagon raises your blood sugar when it's too low. Together, insulin and glucagon are like a perfectly balanced team, always adjusting to keep your blood sugar just right. But wait, what happens if you're stressed out or haven't eaten in a while? Your body needs extra energy to handle the stress or keep you going. That's when a second set of hormones comes into play. Cortisol, epinephrine, or also known as adrenaline, and growth hormone. These are your stress hormones. And here's how they work. Cortisol is your body's stress hormone. When you're stressed, cortisol kicks in to help raise your blood sugar. It's like your body's way of saying, you need energy now. Cortisol works by telling your liver to make more glucose. But the downside is, if you're stressed all the time, high cortisol can mess with your insulin and make your blood sugar stay too high. Well, not great for a long-term health. Epinephrine, or also known as adrenaline. You probably know this one. It's the fight or flight hormone. If you're scared or in a tough situation, epinephrine gets released to give you a burst of energy. It raises your blood sugar by signaling your liver to release more glucose. This is helpful when you need a quick energy. If it's constantly happening, it can be hard on your body. Growth hormone. This one's mostly about helping you grow, but it also has an impact on blood sugar. It makes sure you have extra glucose for energy when you're fasting or working out. Growth hormone stops insulin from doing its full job and raises blood sugar, making sure your muscles have enough energy. So how does it all work together? Let's find out. After eating, your blood sugar goes up, and insulin helps lower it by storing glucose in your cells and liver. When you're fasting or haven't eaten in a while, your blood sugar drops, and glucagon raises it by releasing stored glucose from your liver. When you're stressed or working out, your body needs extra energy, so cortisol, epinephrine, and growth hormones come in and raise your blood sugar to give you that boost. Basically, your body is constantly adjusting blood sugar levels with these hormones, depending on what's going on inside and outside. Now, here's the thing. While stress hormones are super helpful in the short term, chronic stress, the kind that doesn't go away, can cause problems. When your body is under constant stress, cortisol levels stay high. This keeps your blood sugar elevated all the time, which can lead to something called insulin resistance. This is when your cells stop responding to insulin properly. So, your body needs to make more and more insulin to keep things balanced. Over time, this can lead to type 2 diabetes. So, while your body has all these great hormones working to help you in stressful situations, too much stress in the long run can mess with your blood sugar control. So, what's the bottom line? Your body is pretty awesome at keeping things balanced. By managing stress and taking care of your overall health, you can help these hormones do their job and keep your blood sugar just right.
Pyruvate citric cycle. When we eat food, our body breaks it down into glucose. Glucose is then converted into pyruvate in a process called glycolysis. This pyruvate is crucial because it gets turned into acetyl-CoA, which powers the citric acid cycle, the main process our cells use to produce energy or ATP. Pyruvate decarboxylation or link reaction. Before entering the citric acid cycle, pyruvate must be turned into acetyl-CoA in a process called pyruvate decarboxylation or the link reaction. This happens inside the mitochondria. Here's how it works. First, the pyruvate dehydrogenase complex or PDC does the conversion. Second, one carbon from pyruvate is released as carbon dioxide. Third, the remaining 2 carbon piece combines with coenzyme A to form acetyl-CoA. Fourth, in the process, NAD plus is turned into NADH, which will later help produce ATP. In simple terms, pyruvate is broken down to create acetyl-CoA and energy carriers NADH that are needed for ATP production. The citric acid cycle, also called the Krebs cycle or TCA cycle, happens in the mitochondria. It's a series of chemical reactions that turn acetyl-CoA into energy molecules including ATP, NADH, FADH2, and CO2.
These are the steps for the citric acid cycle. Step 1. Formation of citrate. Acetyl-CoA combines with oxaloacetate to form citrate, catalyzed by citrate synthase. Step 2. Isomerization of citrate. Citrate is converted to isocitrate by aconitase preparing for oxidation. Step 3. Oxidation of isocitrate. Isocitrate is oxidized to alpha-ketoglutarate producing NADH and CO2 catalyzed by isocitrate dehydrogenase. Step 4. Oxidation of alpha-ketoglutarate. Alpha-ketoglutarate is decarboxylated to succinyl-CoA producing NADH and CO2 catalyzed by alpha-ketoglutarate dehydrogenase. Step 5. Conversion of succinyl-CoA to succinate. Succinyl-CoA is converted to succinate generating ATP or GTP through substrate level phosphorylation catalyzed by succinyl-CoA synthesis. Step 6. Oxidation of succinate. Succinate is oxidized to fumarate producing FADH2 catalyzed by succinate dehydrogenase. Step 7. Hydration of fumarate. Fumarate is hydrated to form malate catalyzed by fumarase. Step 8. Oxidation of malate. Malate is oxidized to regenerate oxaloacetate producing NADH catalyzed by malate dehydrogenase. These cycles then repeats with another acetyl-CoA. Energy yield from the citric acid cycle. Each turn of the citric acid cycle yields the following high-energy molecules. 3 NADH molecules are produced, 1 FADH2 molecule is produced, 1 ATP or GTP molecule is produced. Since one glucose molecule produces 2 pyruvates, each glucose molecule goes through 2 cycles, doubling the energy output. These molecules are crucial for the electron transport chain, where they help generate a large amount of ATP by donating electrons that power the synthesis of ATP. Regulation of the citric acid cycle. The citric acid cycle is tightly regulated to ensure energy production is efficient and balanced with the cell's needs. Key regulatory enzymes include pyruvate dehydrogenase complex or PDC, citrate synthase, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase. These enzymes are regulated by allosteric control, meaning that molecules like ADP and NAD plus activate the cycle when energy is low, while ATP, NADH, and acetyl-CoA inhibit it when there's plenty of energy. This feedback mechanism helps the cell respond to its energy needs by either ramping up or slowing down the citric acid cycle.
Carbohydrate metabolism disorders are a group of conditions that affect how the body processes carbohydrates, one of the body's primary energy sources. These disorders are broadly categorized into inherited and acquired types. Let's explore some key examples, focusing on their causes, symptoms, and management. First, let's discuss galactosemia, an inherited disorder caused by a deficiency in the enzyme galactose 1-phosphate uridyl-transferase, or also called as GALT. This enzyme is essential for metabolizing galactose, a sugar found in milk and dairy products. When galactose cannot be metabolized, it accumulates as galactose 1-phosphate, leading to severe complications such as liver damage, resulting in hepatomegaly or even liver failure.
cognitive impairment caused by toxic effects on brain tissues, and cataracts due to galactose being converted into galactitol, which damages the lens of the eye. Symptoms typically appear in newborns shortly after milk consumption and include vomiting, diarrhea, and jaundice. Early diagnosis through newborn screening and strict galactose-free diet are crucial for preventing these complications. Next, we have hereditary fructose intolerance, or HFI, another inherited condition caused by deficiency in the enzyme aldolase B, which is vital for fructose metabolism. In individuals with HFI, fructose ingestion can lead to hypoglycemia, or the dangerously low blood sugar levels, and severe abdominal pain, nausea, vomiting, and diarrhea after consuming fructose-containing foods. Also, liver and kidney damage from prolonged exposure to fructose. Management involves strictly avoiding fructose and sucrose in the diet to prevent these symptoms and complications. Acquired disorders such as diabetes mellitus are among the most common carbohydrate metabolism conditions. Diabetes mellitus is characterized by impaired glucose metabolism and is classified into two main types. Type 1 diabetes is an autoimmune disease in which the immune system destroys insulin-producing pancreatic beta cells. Without insulin, glucose cannot enter cells, leading to symptoms like excessive thirst, frequent urination, and weight loss. While type 2 diabetes, on the other hand, involves insulin resistance and relative insulin deficiency, often linked to obesity and sedentary lifestyle. Symptoms develop gradually and include fatigue, increased thirst, and frequent infections. Both types can lead to long-term complications such as cardiovascular disease, nerve damage, and kidney failure if not managed effectively. Management typically includes lifestyle changes, medications, and insulin therapy tailored to the individual. Finally, let's discuss fructokinase deficiency, a rare inherited condition that affects the initial step in fructose metabolism. Unlike hereditary fructose intolerance, fructokinase deficiency is a benign condition. Symptoms, if present, are typically mild and may include gastrointestinal discomfort after consuming large amount of fructose. Management focuses on moderate dietary modifications without the need for strict dietary restrictions. In conclusion, disorders of carbohydrate metabolism, whether inherited or acquired, can significantly impact health. Early diagnosis and proper management are essential to preventing complications and maintaining a good quality of life.
When we consume food, our body breaks it down into glucose, a key energy source. Through glycolysis, glucose is transformed into pyruvate. This molecule plays a pivotal role in cellular energy production as it is converted into acetyl coenzyme A, which is the fuel for the citric acid cycle. But before pyruvate enters the citric acid cycle, it undergoes a process known as pyruvate decarboxylation or the link reaction. Inside the mitochondria, pyruvate is converted into acetyl coenzyme A by the pyruvate dehydrogenase complex. During this process, carbon dioxide is released and energy-rich NADH is produced. Now, acetyl coenzyme A enters the citric acid cycle, also known as the Krebs cycle or TCA cycle, which is a series of reactions in the mitochondria designed to generate energy. First, acetyl coenzyme combines with oxaloacetate to form citrate, catalyzed by citrate synthase. Citrate is then rearranged into isocitrate, preparing it for oxidation. Isocitrate is oxidized into alpha-ketoglutarate, releasing carbon dioxide and producing NADH. Alpha-ketoglutarate undergoes a similar transformation, producing another NADH molecule and carbon dioxide while forming succinyl coenzyme A. This is followed by substrate-level phosphorylation, converting succinyl coenzyme A into succinate and generating ATP or GTP.
Next, succinate is oxidized to fumarate, producing FADH.
Fumarate is hydrated into malate, which is then oxidized to regenerate oxaloacetate, completing the cycle. This last step also produces NADH. The citric acid cycle produces a wealth of energy carriers with each turn: 3 NADH, 1 FADH, and 1 ATP. And since each glucose molecule results in 2 cycles, the energy yield is effectively doubled. These carriers are vital for the electron transport chain, where they drive large-scale ATP production. The citric acid cycle is also tightly regulated. Enzymes like pyruvate dehydrogenase, citrate synthase, and isocitrate dehydrogenase ensure the cycle runs efficiently. Activators like ADP and NAD signal low energy levels, while ATP and NADH slow things down when energy is abundant.